In this video, we are going to discuss or look out for the main differences that exist between glycolysis and gluconeogenetic pathway. In the previous videos, we discussed glycolysis and we established the fact that glycolysis occurs when there is excess amount of glucose in the blood. In that sense, the enzyme or the insulin hormone is released from the pancreas, and that hormone stimulates the enzyme, the key enzymes in the glycolysis, to trigger or to proceed the glycolytic pathway. We established the fact that per 1 molecule of glucose in the glycolytic pathway, there is an equivalent production of 2 net ATPs, 2 pyruvate molecules, and 2 NADH. These are the main products of glycolysis which aid in energy production. We established the fact that the glycolytic pathway involves a consecutive of about 10 steps, of which 3 of them are the key steps. Glycolysis, in converting glucose to glucose 6-phosphate, that is the first non-reversible reaction in glycolysis. Conversion of fructose 6-phosphate to fructose 1,6-base phosphate is the second non-reversible reaction in glycolysis, and conversion of phosphoenol pyruvate to pyruvate is the third non-reversible reaction in glycolysis. Now, these processes are regarded as the rate determining steps in the glycolytic pathway because they are the non-reversible reactions glycolysis that help to control the glycolytic pathway when there is excess production of ATP in the body. Glycolysis basically occurs when there is excess amount of glucose. It occurs in different organs. Basically, in the brain, there's glycolysis in there. Any organ that needs an amount of ATP is vigorously in the muscles. It occurs in several organs because most of these organs, or almost all the organs in the body, needs ATP for its activities. Now, what happens to the body when there is low amount of glucose, or let's say low amount of energy? How does the body maneuver or adapt to such condition? Now, the process called gluconeogenesis is an adaptive pathway which occurs oppositely to glycolysis when there is no amount of glucose or limited amount of glucose. So the gluconeogenesis is an opposing pathway which occurs mostly in the liver, and it occurs when there is no glucose or no ATP in the body. The process occurs under the insulin, or occurs under the influence of hormone we call glucagon, which is also released from the pancreas. Glycolysis occurs under the influence of insulin, but gluconeogenesis occurs under the influence of glucagon. Glycolysis occurs when there is excess amount of glucose in the blood. Gluconeogenesis occurs when there is a limited amount of glucose. Now, let's figure out the main differences that exist between the two pathways. The gluconeogenic pathway is a process by which non-carbohydrate sources or non-carbohydrate molecules are being maxed up through anabolic pathway to produce back glucose. Glycolysis involves the breakdown of glucose to pyruvate, but gluconeogenesis is the process with the opposing pathway of glycolysis, whereby non-carbohydrate sources are being combined or anabolized to form back glucose. So in gluconeogenesis, let's say molecules or precursors like pyruvate itself, you can have some amino acids like alanine, you can have glycerol, and some other molecule like, let's say, lactate. These are molecules which are maxed up in gluconeogenesis pathway to form back glucose when there is limited amount of ATP or no amount of glucose in the body. So let's look at the main difference that exists between the two opposing pathways. So in glycolysis, the steps in glycolysis are almost the same in gluconeogenesis, except 3 steps. So only 3 steps in glycolysis differs in gluconeogenesis because they are catalyzed by 3 different enzymes. 3 enzymes in glycolysis are changed in the gluconeogenetic pathway. The first one is the conversion of glucose to glucose 6-phosphate. This pathway occurs under the expense of ATP, the conversion of glucose to glucose 6-phosphate. So here, the enzyme hexokinase comes into play and adds up a phosphate group to carbon 6 of the glucose. This is the glycolytic pathway So the red side is the glycolytic pathway and the blue side is the gluconeogenic pathway. I'll compare the differences that exist between the two. So this enzyme, the hexokinase, the kinase enzyme always add a phosphate group from an ATP. Every enzyme called kinase adds a phosphate group from ATP to a substrate residue. So in glycolytic pathway, the first step is addition of the ATP or the phosphate group from the ATP to the glucose to form glucose 6-phosphate. This pathway is opposed in the gluconeogenetic pathway because in gluconeogenesis, where you want to produce glucose, phosphate group is added to or is removed from the glucose 6-phosphate to form glucose, and this catalyzed by enzyme glucose 6-phosphatase. Glucose 6-phosphatase. The next reaction which changes in the glycolytic pathway is the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate in glycolysis. This process also occurs under the expense of ATP in glycolysis. So here, the enzyme phosphofructokinase 1 adds a phosphate group from ATP to fructose 6-phosphate to form fructose 1,6-bisphosphate. Basically, add carbon 1 of the fructose 1 or fructose 6-phosphate. This reaction changes in the gluconeogenetic pathway. So here, an enzyme phosphatase comes to play. Know the difference between a kinase enzyme and a phosphatase enzyme. These two enzymes catalyze opposing reactions. So kinase enzymes, as we see here, this is kinase and this is kinase enzyme. These enzymes adds a phosphate group from ATP to to a substrate residue. But a phosphatase enzyme basically removes a phosphate group from a residue. So here, the glucose 6-phosphatase opposes the reaction of hexokinase because it removes a phosphate group from glucose 6-phosphate to form back glucose. Here, the fructose 1,6-bisphosphatase opposes the reaction of phosphofructokinase 1 because it removes a phosphate group from fructose 1,6-bisphosphate to fructose 6-phosphate in the gluconeogenetic pathway. So in the glycolysis, we add ATP by the enzyme kinase. In the gluconeogenetic, we remove a phosphate group in gluconeogenesis to form back fructose 6-phosphate. So these are the first two reactions in glycolysis which differs in the gluconeogenetic pathway. Almost all the steps are the same because the enzymes there or the reactions are irreversible reactions. So the enzymes catalyze both reactions in glycolysis and in gluconeogenesis. The only difference you must see here is that in glycolysis, as we produce NADH from NAD+, in gluconeogenesis, we produce NAD+ from NADH. Once again, in glycolysis, as we produce ATP from ADP, in gluconeogenesis, we produce ATP or we consume ATP into ATP. So bear in mind, in this reaction, we produce ATP in glycolysis, but in gluconeogenesis, we consume ATP. So almost all the steps where ATP is produced in glycolysis, ATP is consumed in gluconeogenesis. The next step, which differs in glycolysis and gluconeogenesis, is conversion of phosphoenolpyruvate to pyruvate. This reaction in glycolysis occurs through substrate level of phosphorylation to produce ATP. So here we produce ATP by an enzyme kinase. So the kinase pyruvate kinase enzyme adds a phosphate group from the substrate, which is a phosphoenolpyruvate, to ADP to produce ATP in glycolysis. And this mechanism is by substrate level phosphorylation, unlike the oxidative phosphorylation which occurs in the electron transport chain. So in this reaction, which also differs in gluconeogenesis pathway, in gluconeogenesis, ATP is consumed. This reaction occurs or is reversed in gluconeogenesis by two-step reaction. The enzyme which converts the phosphoenolpyruvate to pyruvate in glycolysis is opposed by two enzymes in gluconeogenesis. So in conversion of pyruvate to phosphoenolpyruvate, two enzymes come to play to catalyze a two-step reaction to convert the pyruvate to phosphoenolpyruvate. The first enzyme here is pyruvate carboxylase. This enzyme tends to add a carbon dioxide molecule to pyruvate to form oxaloacetate at the expense of energy ATP. Once again, another enzyme which is called a phosphoenolpyruvate carboxykinase removes the carbon dioxide and replaces it with a phosphate group to form phosphoenolpyruvate. So this phosphoenol, the PEP carboxykinase, also convert the oxaloacetate back to phosphoenolpyruvate. So these are the two or three main reactions that differ in glycolysis and in gluconeogenesis. So in the two pathways, glycolysis, three enzymes are replaced by four enzymes in gluconeogenesis. It's very simple. In conversion of glucose to glucose 6-phosphate by hexokinase, it is replaced by glucose 6-phosphatase in gluconeogenesis. In conversion of fructose 6-phosphate to fructose 1,6-base phosphate by an enzyme phosphofructokinase 1 in glycolysis, the enzyme fructose 1,6-base phosphatase converts the phospho or the fructose 1,6-base phosphate to fructose 6-phosphate in gluconeogenesis. In conversion of phosphoenolpyruvate to pyruvate by an enzyme pyruvate kinase in glycolysis, two enzymes, pyruvate carboxylase and PEP carboxykinase, come to catalyze this reaction to convert the pyruvate back to phosphoenolpyruvate. So these are the two main or three main reactions in glycolysis that differ in gluconeogenesis. Three main reactions in glycolysis that differ in gluconeogenesis, and the three main enzymes which are replaced in glycolysis are hexokinase, phosphofructokinase 1, and pyruvate kinase. These three steps are the irreversible steps and also the rate determining steps in glycolysis, which are changed in gluconeogenesis. The other difference is that almost all the steps, as we said here, the steps in glycolysis where ATP is consumed, or the steps in glycolysis where ATP is produced, in gluconeogenesis ATP is consumed. So here the first, where there is the first substrate level phosphorylation, is conversion of 1,3-base phosphoglycerate to 3-phosphoglycerate. Here ATP is produced in glycolysis. That's the red one. ATP is produced in glycolysis, but in gluconeogenesis ATP is consumed. Once again, phosphoenolpyruvate to pyruvate, ATP is produced by substrate level phosphorylation in glycolysis, but in conversion pyruvate to phosphoenolpyruvate in gluconeogenesis, ATP is consumed. So let's look at the key difference that exists between them. So glycolysis is a set of reactions that convert glucose into two pyruvate molecules, but gluconeogenesis is the production of glucose from non-carbohydrate sources. The raw material in glycolysis is glucose, but the raw material in gluconeogenesis is lactate, amino acids, alanine, and glycerol. And most of these are first converted back to pyruvate before they enter the gluconeogenetic pathway, with the exception of glycerol, which can be converted straight to dihydroxyacetone phosphate before it enters the gluconeogenetic pathway. Next, glycolysis occurs in the cytoplasm of the cell, but gluconeogenesis occurs in both the cytoplasm and the mitochondria. Glycolysis is a catabolic reaction because it breaks down glucose into pyruvate, but gluconeogenesis is an anabolic reaction because it converts non-carbohydrate sources like the pyruvate itself back to glucose. Glycolysis is an exergonic reaction because it produces ATP. Gluconeogenesis is an endergonic reaction because it consumes ATP. Glycolysis occurs through a series of 10 steps, but these steps are changed in gluconeogenesis. So these are the key differences that exist between these two opposing pathways. They are opposing because they occur at different times in the same cell or in the same organ, but they can occur at the same time in different organs. For example, glycolysis can occur in the muscle, the same time when gluconeogenesis is occurring in the liver. This is by the Cori cycle. Don't forget to type. Thanks for watching. Don't forget to like and subscribe to the channel after watching. In the next videos, we are going to look at the electron transport chain and how this produces energy equivalent from glycolysis, from the Krebs cycle that we looked in the previous video, and also conversion of pyruvate to acetyl CoA are used up to produce ATP by oxidative phosphorylation. Thanks for watching. Don't forget to subscribe.